Great, thank you so much, Michelle. And it's lovely to see you all here today, and it's nice to see some familiar faces, and just thank you so much for coming and kind of joining us on this day. So I'm going to start off on a lighter note.
It's a brobrig moonlicht, nicht the nicht this morning. There's a bonny wee lassie out there, hootsman the new.
You mind sitting down there, please, Mr. Hackman?
Beg your pardon for lapsing into the vernacular, but the young lady did say you were a Scottish gentleman.
Well, not all Rob Roys. Have your card, please.
By all means. I'm ready when you are, Squire.
Yes. Hold out your hand, please. Now, this won't hurt. You'll just feel a slight prick on the end of your thumb.
Well, I'll bid you good day then. Thank you very much. If you want any more, don't hesitate to get in touch with me.
Where are you going?
Have me tea and biscuits.
Thought you came here to give us some of your blood.
Well, you've just had it.
That's just a smear.
It may be just a smear to you, mate, but it's life and death to some poor wretch.
I've just taken a sample to test.
A sample? How much do you want, then?
Well, a pint, of course.
A pint? Have you gone raving mad? You must be joking.
A pint is a perfectly normal quantity to take.
You don't seriously expect me to believe that? I mean, I came in here in all good faith to help me country. I don't mind giving a reasonable amount, but a pint? That's very nearly an armful. I don't mind that much, but I'm not having it up to here. Sorry, I'm not walking around with an empty arm for anybody. And after all, a joke's a joke and all that.
There we go. So, I mean, this is, I didn't know about this comedian before he came to the UK, but I definitely got to know him working in the research group. And today I'm here to explain a little bit about what we do with that armful of blood that we take every time. So, and the topic today is proteomics. So I'm going to start just with some basics. So this is an image that kind of shows how DNA kind of provides the code or the recipe for us to produce proteins, right? So at the top, we've got the DNA molecule, and at the bottom where it says polypeptide, that's your protein, right? And when we study the DNA, we talk about genomics, right? And so if we study proteins, we talk about proteomics. So how many proteins are there? So, I mean, this recipe book of ours is extremely comprehensive, and it's got about 3 billion base pairs. And essentially what that means is if you lined up all the little constituent parts of your DNA in one straight line, it'd be 2 meters long, right? So this is a huge, huge molecule. It's got all the instructions for all your proteins. And we roughly think there are about 20 to 25,000 proteins that can be made with your DNA molecule, right? So what do these proteins do? So proteins have a variety of functions. So it can be, you know, we've heard today about enzymes. So enzymes are proteins that kind of help reactions happen in the body. You know, when you have an infection and your body produces antibodies, those are types of proteins. Kind of what provides the scaffolding in a cell to kind of keep it from collapsing in itself. Those are structural proteins. We got proteins that transport molecules in and out of a cell. And then we have proteins that kind of speak to each other within the cell, kind of what we call cell signaling, to kind of tell the cell to increase the expression of a protein or reduce the expression of a protein. So these are kind of broadly speaking, the functions of proteins. So how do we normally measure proteins? Well, things that you'd be kind of exposed to, you know, if you interact with a GP and have tests done. So, you know, an ELISA is a type of protein test that essentially you design an antibody that sticks to a particular protein, and then you essentially you give your blood, you add the antibody, and you see if it sticks. If the protein is present, it will stick, and then there's a little marker on it that kind of lights up, and that allows you to see whether or not that protein is present in your blood. The trouble with the first one is that you can only really look at one protein at a time. So you really got to sit down, you got to design, and you know, otherwise you get a very messy signal. So you can only look really at one at a time. So the second approach is a classic approach, which is kind of called the Western blot. And essentially you take, let's say you have a whole bunch of proteins in a sample and you want to have a look at kind of, you know, what's in the sample. You stick it in some gel, and you pass some electricity through it, and it kind of separates out into these bands. And then you can light them up, and you can kind of tell roughly, what's present in the sample. And then we have a more, like a newer technique that's come around in the last decade or so, which is the mass spectroscopy. So this is a very clever technique where essentially they fire proteins at extremely high speeds through a magnetic field, and depending on kind of the chemical properties and physical properties of the protein, it will deflect differently and hit the detector at a different point, and then they can deduce what protein it is. Very clever. But there's a new kid on the block, and one of these companies that has designed this new kit, this new technology, is a company called Olink, which is they're based in Uppsala, Sweden, and essentially what they allow us to do, what this technology allows us to do, is to look at thousands and thousands of proteins all at once, in one go, on a tiny little bit of light. And essentially, and I've got a little video which I'll show, but just to kind of get an idea, it's like, how does this technology work, right? So we're talking about proximity extension assays, but don't worry about the terminology. Essentially, what you've got to think about is, you get this antibody pair, so these two antibodies that are made to stick to a particular protein. And these antibodies have got this little bit of DNA on that we've made, that we've stuck onto the antibody. When these two antibodies click into the protein, these DNA strands lock in, and that provides a barcode and tells you, okay, that protein is there. And then we essentially use established gene DNA reading machines to kind of read those barcodes. So I'm going to give you a video.
Proteins are key biomarkers for disease and drug responses, but measuring them is usually difficult. Traditional assays for proteomics have major trade-offs for multiplexing, specificity, sensitivity, throughput, and sample volume. However, Olink's technology is a game changer. It combines the advantages of genomics with advanced proteomics. Here's how it works. Olink's proximity extension assay uses two matched antibodies per target antigen. Each antibody pair is labeled with unique DNA oligo barcodes. When the antibodies bind to the same target protein in solution, the DNA oligo strands anneal, with enough stability to enable DNA extension. The DNA barcode is then amplified, and the resulting amplicons are measured by qPCR, or next-generation sequencing, for absolute or relative quantification. Olink's scalable assays speed up discovery, development, and clinical studies. They provide an unparalleled combination of multiplexing, specificity, sensitivity, throughput, and sample volume, delivered in a convenient output. Analyze up to thousands of markers simultaneously, using as little as 1 microliter of a sample.
Right, okay, so hopefully that kind of gives you an idea of how this technology works. So now that brings me to something that Kalina actually mentioned, and this is, how are we making this Olink experiment possible? So you kind of have a bit of a background, proteins, where they come from, why they're important, what they do, and how we detect them. But now how did this whole project of proteomics come to be? So thanks to an amazing partnership between GSK and the University of Oxford, we now have, as of 2022, the Institute of Molecular and Computational Medicine. And fortunately, one of the major themes of this project is research in Parkinson's disease. And so through this partnership, we've been able to get funding to do proteomics analysis, or using the Olink platform, on a lot of samples, so hundreds and hundreds of samples. So what is our focus? Why are we doing this? What are we looking for? So we're looking for biomarkers. So you may have heard this term before, and it just kind of sounds fancy to call it a biomarker, but it's essentially just a marker, right? A marker of a disease. And we can use biomarkers for a whole bunch of reasons. But our objective is to find biomarkers in Parkinson's disease. So the types of biomarkers that we have, this is just kind of a selection of of them, but the ones that we are really interested in, the diagnostic biomarkers, all right, essentially establishing whether or not a disease is present, and prognostic biomarkers, telling us whether and how a disease will evolve over time. So, as a practical example, right, let's say you were diagnosed with REM sleep behavior disorder. And as you know, there's a very strong link between RBD and Parkinson's disease. But you would probably want to know, if you're on that journey to Parkinson's, whether or not you're one of the lucky few that doesn't develop Parkinson's, or, when this point is going to come, in terms of planning your life ahead. Because some folks can have RBD for decades and nothing happens, and others have it for a couple of years and develop Parkinson's. So if there was a diagnostic biomarker that could tell us, okay, now this is when you develop PD, or the PD has already manifested. You're starting to lose dopamine neurons. Is there some kind of marker that can tell us, right? So we can call that the diagnostic biomarker. Or conversely, let's say you've got Parkinson's or disease already, and you want to know, what your journey of Parkinson's is going to look like. So, there's been a lot of discussions about, clusters and different clusters of Parkinson's, because as you know, everyone's Parkinson's is different, right? Everyone's journey with Parkinson's is different. Some people progress very quickly, some people progress slower, some people have lots of non-motor symptoms, some other people lots of motor symptoms. So kind of being able to figure out kind of what trajectory you're on would provide a wealth of information to patients. So that's kind of where we're looking for what's called a prognostic biomarker, right? That can help us predict the future, essentially. Or at least give you an idea of what to expect. So that kind of gives you an idea of why biomarkers are important in a practical sense for you, right? But for us, in terms of drug trials, this is also an extremely useful tool. Because this is, let's say, a hypothetical drug trial, right? This is a drug trial in Parkinson's today. And, so at the top, you've got the placebo, and at the bottom, you've got drug X that they're trialing in the drug trial. And normally what we would do at the moment is, because we don't really have a robust, reliable biomarker in Parkinson's disease that kind of allows you to monitor how the disease progresses, we use clinical scores, so the UPDRS. So, when you come to clinic and we make you, do this and open your hand and tap your foot, we're essentially trying to gauge the severity of your motor symptoms. But as you'll know, essentially what we want to try and do is see if there's a difference, right? So does the score for somebody on the drug not progress as much, right? Is the drug having an effect in terms of slowing down the Parkinson's disease? So we're trying to find that gap, right? So where the placebo your progression just continues, but when you're on the drug, the progression slows down. And we use this clinical motor score to track that. But as you'll probably know, this motor score is not the most reliable thing. It kind of depends who's doing it on the day, like how you're feeling on the day, if you're able to relax, because sometimes we ask you, try and relax your limbs, imagine you're on the beach, and sometimes it's difficult, and then you tend to score higher if you're a bit stressed. So these kind of things can influence the score and doesn't really give you an accurate kind of estimation of whether or not the disease is progressing. So clinical motor scores aren't great. So we would love to have a situation where we have a protein that can tell us whether or not your disease is progressing, right? So there are other diseases where we're able to do that, and we would love to be able to do that in Parkinson's disease. So find some kind of biomarker that can tell us, okay, has the Parkinson's really slowed down? Has the drug really worked versus the placebo? So you see, this kind of explains the importance of this in kind of drug research and why there's so much investment and interest in this. So where are we now with our current proteomics project? So we essentially have two kind of arms of the project that are running simultaneously. By far the biggest one is the serum experiment. So as you can see, we have 1,912 samples. It's huge. It's a huge experiment. So and then we have a smaller cerebral spinal fluid experiment. And kind of this gives you just an idea of where we are on the journey of this project. So, the data's in, we've got it now, so now it's just about analyzing this. But that is easier said than done when we're talking about millions of data points. So, over the next couple of months, this is kind of really what we're going to sink our teeth into. So, to recap, so some key points. So So proteomics, as we now know, so proteomics is the study of proteins, right? And we use modern techniques like Olink, allow us to study these proteins and thousands of them at a time. Then the other key concept, biomarkers, right? Prognostic and diagnostic biomarkers, being able to tell you whether or not a disease is present, how the disease is going to progress over time, and using these biomarkers in drug trials to really give us a reliable mark of whether or not a drug is working. So in summary, we take your blood, a bit of that armful of blood, we run it through this machine, we get a whole bunch of data. With that, we are going to try and find a blood signature of Parkinson's disease. I just want to thank you. I just really want to thank Michelle and just the wider group and the folks from IMCM for kind of making this all possible. And that's me. Thank you very much.